Hello everyone. With the discovery of DNA, there was another concept which came into existence that was about central dogma. Central dogma is a process in which the instructions in the DNA are converted into functional mRNA, which codes the information to form proteins capable of expressing the phenotype. So, according to central dogma, there are basically three things which occur. First of all, there is presence of a DNA. This DNA will copy itself and form multiple DNA units, because every time a cell division is occurring, some amount of DNA has to be forwarded to the next cell as well. And in order to maintain the normal 23 or 2:1 ratio of a cell, the genetic material must duplicate itself. Here, genetic material duplication means one of the strand of DNA is present. Let me just make it once again. If one of the strand of DNA is present, then we can make another strand which is complementary to it with the information of nucleotides we already have. And now this DNA will be again separated. So half of the chromatid will move to one of the cell and another half to the another cell. So with this information keeping in our mind, one DNA before its division or before the cellular division of one of the cell, the nuclear material, it duplicates itself. It means it forms both the sister chromatids, and after the formation of the sister chromatids, the DNA it splits into two. Again, half of the chromatid it will be going to one of the cell and another half to another cell, and as a result we see the formation of two new cells having half of the genetic material, but the ploidy is definitely 2n. Just the genetic material, little amount of genetic material, just half of the chromatid has went to one of the cell. It is not haploid condition, it is just half of the genetic material has went to one of the chromatid. 2n number will be retained during this process. Now after that, we see one more thing happening. Once the DNA is formed, after DNA formation, this information of the DNA is transcribed in order to form an RNA. After the formation of RNA, this RNA is a specialized RNA, which is mRNA. The entire information of the nucleus or how the genetic material is coded in an messenger RNA, and that messenger RNA further goes to the cytoplasm, and over there it forms the proteins. Now, whenever any character is expressed in us, it is the proteins who are expressing it. So, this entire idea was developed by the discovery of, or by the time the discovery of DNA occurred. So, this all information which we discussed, it was all given up by Francis Crick, who was the person who discovered the DNA as well. But by the late 19th century, this like within 20 to 30 years, 1953, the discovery of structure of DNA occurred, and within 1970, another information came into account. The information was that the RNA viruses, they can also transform their information in the genetic code in the form of DNA. And with this information, a great revolution took place. It was that we till now we were aware that the information is flowing in a unidirectional way from DNA to RNA and from RNA to a protein. But now there was a change. Now DNA was, or RNA was also capable of forming DNA. So this became a very revolutionary idea. So now the central dogma was changed, and we, as the information which we all had, that there is unidirectional flow of hereditary information from DNA to RNA, mRNA, and then to protein. Now it was that it was said in a little with a little modified way. Let's see how. DNA to DNA formation is known as replication. Replica formation of a DNA is done with the help of nucleotide sequences being formed. If we forward one of the cell with just half of the DNA structure or just one of the strand of the double helical structure, the another half of the strand can be formed very easily with the nucleotide sequences of the previous parental strand. That information is only utilized in order to form a DNA again, or the another half of the DNA. When two cells are dividing, they are having only half of the information, not half of the information, full information, but just one of the strand. I hope it is understood. Full information, but half of the strand. Another half is formed, and that is known as duplication. In the S phase, synthesis phase of DNA, or in the mitosis, you see a S phase. In a cell cycle, presence of S phase is there. That is the phase where duplication of genetic material occurs, which means that half of the single-stranded DNA will form another half, so that complete information is present. That is replication. Next is from DNA to RNA conversion. This conversion is known as transcription, where one of the strands com- or information of one of the strand is read but with replacement with uracil. Everything will remain the same if it is A, T, C, G, the strand, if it contains this kind of information. The mRNA will also contain the similar kind of information, just T will be replaced by uracil. So wherever you can see T, it will be replaced by U. A will be A, but T will be U. C will be C, but G will be G. Wherever you see T, it will be replaced by U. So this is about transcription. Once the transcription is done, there is another one thing which happens. It is reverse transcription. Transcription is taking place in normal DNA to RNA conversion in the organisms who have genetic material as DNA. But the retroviruses, they have this another technique of conversion of their genetic material into DNA. So reverse transcription means that uracil will be replaced by thymine, and this takes place when the retroviruses, they infect any individual, they change their genetic material, and they completely modify the DNA of the parent, or they just modify their own DNA in such a way that uracil is replaced by thymine and get incorporated into the nucleus or nuclear matter of the cell which they are trying to invade. This is a special case for retroviruses. And the next was the linear flow. In the linear flow, again, translation occurs where, which takes place in the cytoplasm, where RNA is used, or mRNA sequence is used in order to code for a particular protein. So suppose five proteins make a five mRNA nucleotides will make one of a protein. So all of them, they will be coding for five different amino acids, and each of the amino acids, when they are present or sequenced one after another, then they form a viable chain, 5 prime to 3 prime viable chain, and that chain will be capable of forming the protein which we require in order to express a character. Suppose the character is eye color of an individual. So as an end product of central dogma, we now have an idea that DNA can self-replicate itself. DNA can form an RNA with the help of transcription. RNA can form a DNA with the help of reverse transcription, and RNA can also form proteins which express a character by translation. And with this all information into account, molecular biology gives information or gives us an idea how genetic transformation may have occurred during the course of evolution.